Non-invasive transmission of sensor motor information in humans using an EEG/focused ultrasound brain-to-brain interface. Transcranial focused ultrasound modulates the activity of primary somatosensory cortex in humans. Improved methods of non-invasively modulating human brain function are needed. Here we probe the influence of transcranial focused ultrasound, TFUS, targeted to the human primary somatosensory cortex, S1, on sensory-evoked brain activity and sensory discrimination abilities. The lateral and axial spatial resolution of the TFUS beam implemented were 4.9 millimeters and 18 millimeters, respectively. Electroencephalographic recordings showed that TFUS significantly attenuated the amplitudes of somatosensory evoked potentials elicited by median nerve stimulation. We also found that TFUS significantly modulated the spectral content of sensory evoked brain oscillations. The changes produced by TFUS on sensory evoked brain activity were abolished when the acoustic beam was focused 1 centimeter anterior or posterior to S1. Behavioral investigations showed that TFUS targeted to S1 enhanced performance on sensory discrimination tasks without affecting task attention or response bias. We conclude that TFUS can be used to focally modulate human cortical function. Brain-to-computer interface, BCI, techniques detect neural signals associated with brain function and translate them to computer/machine control commands. Conversion of the commands to stimulate specific brain regions of other individuals allows for the formation of the brain-to-brain interface, BBI, that directly links the specific brain activities among individuals/biological entities. In this context, brain stimulation modalities controlled by a computer are considered as the computer-to-brain interface, CBI. Capitalizing on internet connectivity of computers as an intermediary, BBI has the potential to transmit neural information over long distances with a wide information bandwidth. Direct cortical recording/stimulation achieved by implanted electrodes has been used to obtain neural signals, as well as to deliver stimulation to the designated brain regions among rodents, 1, 5, and non-human primates. 6, 10. These implantable devices can be used for achieving BBI, however, the risks associated with the surgical procedure for the electrode implantation limit its applicability to humans. Therefore, to be compatible with experimentation in healthy humans, non-invasive procedures need to be considered for both BCI and CBI modalities. Various BCI systems have been developed and become widely available based on non-invasive neural recording/neuroimaging techniques such as surface electroencephalography, EEG, 11, magnetoencephalography, MEG, 12, and functional magnetic resonance imaging, fMRI. 13. For CBI portion of the implementation of BBI, non-invasive brain stimulation techniques, such as transcranial magnetic stimulation, TMS, 14, 15, or transcranial focused ultrasound, FUS, 16, 18, are available for translating the digital information from a computer to stimulate a certain region of the brain. Since the initial theoretical conceptualization, 1921, we have witnessed the actual implementation of the non-invasive BBI techniques in recent years. For example, we have shown that human intention, as detected by the EEG steady-state visual evoked potentials, SSVEP, can be translated to stimulate the rodent brain of the tail motor area using transcranial FUS, 22. Li and Zhang have reported interfacing human brain function, driven by the SSVEP, to control insect behavior by electrically stimulating their antennae, 23. Grau and colleagues used the binary coding of motor imagery-mediated EEG potentials from a human subject to remotely actuate a TMS device that stimulated the visual areas of another human individual, thus, granting the transmission of motor imagery intention between the subjects, 14. In the same year, Rao and colleagues used motor imagery EEG signals from an individual playing a simple computer game, and stimulated the motor cortex of another subject to push a fire button, who was simultaneously playing the same game using TMS. 15. The same group further applied a similar technique, combined with a traditional motor task, manipulation of a computer mouse, to allow mutual interactions between two individuals in solving a 20 questions game. 24. TMS is an established non-invasive brain stimulation modality, and is the preferred choice as a non-invasive CBI. Although the affected area by the TMS stimulation is on the order of 0.5 to 1 centimeter, 25, better spatial selectivity is warranted. Also, TMS is often accompanied by undesired tactile sensations from the scalp, through activation of skin tactile receptors, and loud clicking sounds, due to the pulsed application of strong magnetic field. 26, 28, which make a confounder-free study design difficult to attain. In addition, one must consider the presence of mutual interference/distortion/interaction of the magnetic field, especially when multiple TMS coils are to be placed in close proximity and actuated simultaneously. 29, 30. Transcranial FUS technique, on the other hand, uses a highly focused formation of acoustic pressure waves to stimulate the neural tissue including the deep brain structures, and has small neuromodulatory areas, on the order of millimeters in diameter. The acoustic energy can selectively reach deeper target areas than TMS. Although the exact mechanism behind the stimulation is not clearly elucidated, the effects of FUS have been associated with mechanical coupling between the acoustic pressure waves to various neural components, which ramifies into changes in membrane capacitance, mechanoreceptor-mediated action potentials, neuron-glia interactions, and/or neurotransmission. 16. 17, 31, 33. This is contrasted to the mechanism of TMS, also not clearly understood, whereby the application of a strong magnetic field induces electrical currents in underlying neural tissue. FUS has recently been tested to elicit tactile sensations from the hand area by stimulating the primary somatosensory area, SI, of the human brain. 16. It has also been used in modulating visuomotor function of non-human primates. 34. As well as in changing sensations associated with passive tactile stimulation. 18. Due to a small footprint of the FUS transducer, and the non-electromagnetic operation in ultrasound sonication, mutual electromagnetic interference does not occur between multiple transducers. Recently, we have shown that both SI and secondary somatosensory area, SII, on the same side of the brain hemisphere can be successfully stimulated using two separate FUS transducers. 35. Therefore, selective and separate stimulation of a brain region in the left and right hemisphere can be readily performed, all of which indicate the utility of FUS as a new mode of non-invasive CBI. In the present study, we detected the neural function associated with two-directional, right or left, motor imagery choices using EEG-based BCI from human participants, as a sender, and relayed the information to selectively stimulate either the right or the left SI of the hand from another individual, as an information receiver, by independent operation of a two-set of FUS transducers. Subsequently, the timing of tactile sensation, elicited in either the left or right hand, was reported through finger tapping of the corresponding hand. The region of FUS stimulation was image-guided based on individual-specific functional neuroanatomy. Two individuals were separated by a large distance, 30 km, and information packets were transmitted over the Internet to achieve BBI implementations. The EEG data for the BCI procedure is not currently available for public access due to privacy concerns, as enforced by the Institutional Review Board of Korea Institute of Science and Technology, KIST. However, individual, case-by-case access to the data is available via request, and interested parties may request the data to Research Support Department of KIST, shko@kist.ri.kr, with the approval number 2014-008. All other relevant data are within the paper and its supporting information file.